Welcome everybody. It's my pleasure to invite you for the second day of LifeScience Open Space event. I'm glad you are here. As you see, we can manage to be here because a lot of people will come later just for their sessions, and this is their loss because this is one of the most interesting part of the event. It will happen now. As you know, yesterday we had to replace two persons our keynotes because of the snow. I'm correct, yeah. But before we start, this is the second day of the LifeScience Open Space event 2023. Today there are still two sessions on two rooms, and also we are including today the third place, which is Atrium, where we start about 11 o'clock presentations from Hackathon, which happened yesterday and before yesterday. Hackathon, can remind you that this is a hackathon for active healthy aging. So this invented by us. Few words about today's. Again, we are still voting. You are still supposed to vote for the best presentation, most compelling, best presentation, most compelling, most effectively activating you, inviting you for collaboration, because this is the main purpose of those speeches here, of those presentations. This is point one. Sessions on both sides. Or in the second room, Enterprise B, there will be startup scene. We have to, of course, remind about our partners, sponsors: Kraków Technology Park, LifeScience Kraków, Sano, behind this sense, and Kraków City. After the sessions, we'll be gathering again in the atrium for the final session, when we will organize kind of wrap-up and discussion. That will be very interesting discussion, very interesting panel, because we are going to reverse the order. As you know, always we invite experts to come to the panel sessions and we ask them questions. This time, this will be just opposite. This will be the experts asking questions, and this is a session we named "Questions That Matter," and it's very interesting. It will be very interesting actually to engage you in this discussion. What kind of questions matter? Now, without any longer, I would like to invite Ingmar Hoer, founder of CureVac. Who told us a story about RNA revolution, and that would be a kind of story, I believe, about the entrepreneur and the way you started and didn't win the Nobel Prize. Okay, here's yeah, come on. Just remember, I just wanted to remind you that there is this type of time for photos. Okay, once more. Okay, don't forget about that.
Okay, thank you very much for having me here one day later. Anyhow, it's very nice to be in Krakow again. It was the 90s. When I was a young boy, after school, I went by bicycle to Krakow. And it was really a nice thing. Yeah, I love Poland somehow. I always come back to Poland because of these times, extraordinary times. Thank you very much. So let's start on the RNA revolution. Is it going forward? Okay, it's going forward. Yeah, you know, the Nobel Prize was given this year to Katalin Karikó and Drew Weissman, one entrepreneurs working on the RNA subject. And actually their technology saved the world because it was the principle of the corona vaccines, their technology, the RNA modifications they did. Well, RNA, let's come to RNA itself. It's a really nice, interesting molecule. A lot of people forgot about it, especially in the 90s. Nobody was working on RNA, but it's the oldest molecule in the world. The first biomolecule was RNA, indeed. That's really interesting. Why nature was developing this kind of molecule? And you know, if you work with RNA, it seems RNA is very unstable. When I was in my lab doing the PhD, it was a hassle to work with RNA. And scientists, they believe RNA is very unstable, you have to avoid it to work with RNA. Why is like that? Because I'm coming from a chemistry department, so therefore chemically RNA is very stable. It's the most stable molecule actually, more stable than proteins or than even DNA. You can boil RNA, you can heat it up 100 degrees and nothing happens to RNA. So therefore chemically it's really stable. And that's the reason why nature developed molecules destroying RNA called RNases, destroying enzymes of RNA. And RNases are all around, you know, if you speak, there are RNases, and RNases destroy the RNA. And that's the artifact seeing in the lab. If you work with RNA, a lot of RNases are around, therefore RNA is going to be really easily destroyed. And therefore, you know, this is the kind of the thing you have to think about. It's the oldest molecule in the world, the most stable molecule we have in biochemistry. So that was the beginning of the evolution in the world. RNA was there, DNA, you can see the double strand DNA. We know the DNA, it's on our chromosomes, chromosome material. And people used to work with DNA for a lot of times. You know, gene therapy, for example, in the 90s, and there were a lot of problems using the gene therapy approach because DNA is very stable. It has to be inherited from generation to generation, therefore it's a stable molecule. And there are not that many DNases, so destroying enzymes of DNA, then RNases destroying enzymes of RNA. So therefore people working with DNA a long time, but DNA, it's very hard to control. Once DNA is in your body, it's expressed, so proteins are made for kind of weeks. You don't know how long exactly proteins are made from that. Using RNA, it's more easy because RNA is destroyed in a couple of hours or days as you want. You have a mechanism to stabilize RNA, therefore you get one-shot RNA, you know, for 2 days there's an expression, then the RNA is gone. That's not the case with DNA. You never know what happens to the DNA injected into your body. So again, RNA is heat stable, it's well soluble, and you can easily produce large quantities of RNA. That's also possible. So actually, I'm from Tübingen, that's the south part of Germany, and actually RNA was discovered, and DNA as well, in Tübingen by Friedrich Miescher at that time, in the 80s, in the 1800s. He didn't really know what he detected, so it was long before the code, the genetic code, and the things like that. He was just thinking, if something is not a protein, but it's in every animal, I find it, so it must be very important. That was his methods at that time. And it's interesting that in Tübingen, the RNA revolution was started by our company. So we got entrepreneurs, and interestingly, IT entrepreneurs like Dietmar Hopp, the SAP founder, or Bill Gates, Microsoft, because somehow it seems RNA is a kind of IT molecule. It's an information molecule. It's called messenger RNA. Messenger gives you information to the cells. And that's interesting. Long before AI things, it was already clear to me that it's quite easy to speak with the body, to talk the body what to do if there's a health issue in the body. So that's Tübingen. You can see it's a very nice medieval town, and on the left side is the new technology park, like here, 20 years ago. And also there were a lot of publications on this technology. And these were actually our first labs at the university, where we could use old chemistry faculty labs, where we started a business due to a grant to our Baden-Württemberg. So the state of Baden-Württemberg gave us some money to start a business. Otherwise, it would have been very tough. Actually, we were quite young guys from the studies, you know, and going to the VC companies, they say, well, who are you? Why you think you want to revolutionize the world? How you dare to do that? So therefore, it was quite impossible to get money funding. So this kind of situation at the university, our first employee in our lab, in our conference room in front of the elevator. So it was always blink, blink, blink when we were talking, people were coming out and in. Yeah, and then we moved in 2003 to the technology park in Tübingen. We were the first ones in the technology park because it was a very dark time. A lot of companies went insolvent, so we were the only left. And our company car as well, we're very proud. I guess we are Germans, we are proud on cars, so therefore our first company cars. So therefore, CureVac developed quite nicely, and it's also noted on the NASDAQ in New York. Yeah, and then we got our first money from a small venture company in 2003. Our employees then 2090s, now they're double or triple or fourth time, 5 times size like this. Many more people are engaged here. Of course, we are very proud that we have a lot of collaborations with a lot of companies, also with the Bill and Melinda Gates Foundation. So that's quite interesting. And it's a really neat that we also get know-how from other parts of the scientific community. Well, I already told you, I think the most important thing for us was that we implemented the pharmaceutical production, so the GMP production, good manufacturing practice production. That's not easy because you have to maintain all the things like the pharma industry, like the big pharma industry. And we were quite a small company, not knowing exactly what to do. And I think this was a really strong impact also for the others following us. Therefore, we invest a lot of money to get this approval and to start clinical trials, because without clinical trials, just having mice data, it tells nothing. So people really want to see clinical trial data on humans. So this was the most important impact in our development, that we could start clinical trials with humans. So we did a lot of trials already in flu, in polio, and of course COVID as well. And let's come again to AI things. It's about information. So it's an information-driven pharmaceutical. It's from nature because we don't modify RNA. Now we have seen that it's needed to modify the RNA, like BioNTech and Moderna. Pseudouracil is a new building block, artificial building block in RNA. But anyhow, it's kind of a software approach. We do that. And once again, from school, you maybe know DNA, chromosome material, then there's the messenger, the RNA, and from this messenger, proteins are made. And that's a lot of important patents. There's also kind of struggles with the others, BioNTech and Moderna, about patents we submitted already in the beginning of the 2000s. A very important, and you can just see the infringement battle with BioNTech. It's on this patent. It's a patent called GC enrichment. GC means guanosine, cytosine. They are 2 building blocks of the RNA. You can see these are the adenine, thymine, guanine, cytosine building blocks. Adenine and thymine are binding with 2 hydrogen bonds, and guanine, cytosine with 3 hydrogen bonds. So that means the more hydrogen bonds, the more stable the RNA is. And that's the patent. We found out that we should use GC, guanosine and cytosine. The more we have of this basis, the more stable the RNA is. And that's the patent. Where's the conflict about? And you can see that's the kind of AI language of the genetic code. Triplet is coding for 1 amino acid. Amino acids getting a protein. And you can just use this scheme to do any protein you want. You can code anything what you really want on RNA. So it's an algorithm you can use. What we implemented were some called of untranslated regions. So regions that are just for stabilization of the RNA on 5' on the one hand, and 3' on the other hand. That the RNA is kind of complexed and stabilized with proteins which are available in the cell. So this is a kind of natural stabilization process. We don't use any modified bases like Moderna and BioNTech. And this is the technology of Kati Karikó, the Nobel Prize winner. She was just replacing uracil, that's a building block of RNA, with pseudouracil. It's a small difference you can see here. There's 1 more N included in that, or the N is switched from one place to the other place. This actually is an interesting technology because it uses the immune response. So we were aware about it already 15 years back when she was applying also in CureVac. We should use her patent and we should use her technology. And she was using that because the immune response was somehow declining. So when you inject RNA in the body, RNA is a danger signal. So if there's RNA in the body appearing, the body gets into danger. So you get huge dangerous signals, huge immune response. And the reason why it's like that, because body is fearing there's a virus attacking it. There are a lot of RNA viruses around. Therefore, RNA gives you a danger signal in the body. So if there's some RNA attending the body outside of any cells, body gets in a danger. And what Carico did, the pseudouracil is somehow missing the danger signal. So the danger signal is reduced. And that was the reason why CureVac failed in the COVID vaccines, because CureVac had to increase the dosages to get an immune response. And the higher the dosage, the more the immune response was there, and an artificial immune response against everything. So CureVac was forced to reduce the doses to get a proper immune response. And the dosage was too low to deliver the information to the body. As I said, it's a kind of information. The body needs information. It was a too low doses. It couldn't really increase the dosage. Whereas Moderna and BioNTech, they could use 4 times more dosages than CureVac. And this was enough to induce the immune response and was enough signal, and there was really no side effects on that. So that was the reason why CureVac failed in utilizing the COVID vaccinations. And that's the reason why BioNTech and Moderna did so well in that. And that's the reason why Carico got the Nobel Prize this year. You can see it here, what I told you. So Moderna already used 100 microgram, BioNTech 30, and we just 12 microgram. It's really the highest dosages. Otherwise, it's too high, and it's not really getting a lot of side effects higher than that. So that's the reason. Well, again, we are really proud that we got the first GMP approval like that. And of course, there are a lot of preclinical and clinical work. We started with oncology. That was more important to us in the beginning, and then we switched to COVID-19 vaccines. You can see there were a lot of trials going on, but we didn't succeed. We couldn't get the approval there. So again, what's also important to know that the production is always the same. It's just the sequence you change. So the production process remains always the same, whether you do a vaccine, whether do you a cancer vaccine, COVID vaccine, or you do some different thing, or an enzyme like that. It's always the same process, and that's the beauty in it. Because RNA is like software. Go ahead. Why is it not going ahead? Yeah, okay. Another nice thing, because I told you already, it's a quite of easy process and always the same process. You can have a kind of printer of RNA. That's really interesting. It's a kind of device. You can just type in the sequence you want to be printed out, and the printer is printing out. You have to put all the ingredients, enzymes like that, into the printer, and then you get your RNA constructs in the end. And one day you make an influenza vaccine, the other day you make a COVID vaccine, the third day you make a prostate cancer vaccine. That's the thing, and you can really have it kind of mobile station. So if there's an outbreak somewhere in Uganda, in Africa, a new COVID outbreak or a new other disease, you can just implement a machine there, and you get it printed out where the spot happens of this outbreak. So that's the print box. And we invented that, and we are very happy that we could collaborate with Elon Musk from Tesla, because Tesla is building some very important building blocks of this machine. So therefore, we met also Elon Musk coming to Tübingen. He's really also interested into this. And when we started the Tesla collaboration, we met him in Palo Alto, and he was asking me why I should work with you. We do cars. We want to work with BMW and this kind of, why I should work with CureVac. We say, well, Alan, it's like flying to the moon with us. We do the same, like you, in your vision, because we revolutionize medicine. And therefore, we need a printer. And you have the technology to provide us with a printer. So in the CureVac printer, there's Tesla technology included there. And I think that's the reason. And we are just the only project outside of car automotive industry. He's not dealing with anybody else than with CureVac, because he was really interested in fulfilling this vision. So that's a really interesting thing, and you can see just on the picture, it can be somewhere in the jungle. You can have a solar energy module, and then you can independently produce RNA. Yes, and then the pandemic, we know about already that. And again, you can really do, and you can see here depicted the proteins, they're really different, and that's the reason why it's so difficult to work with proteins and to make proteins, because they're very different in the size and also in the shape. RNA is always the same, as I said, and why not use the body produce the proteins and not the machine? So let's coming to the future, and I think it's a real disruption we can see here. Because now we are in a situation that we can talk to the body. We can have a recipe to the body, saying the body, well, you know, you might get cancer, so we can have a biopsy of the cancer tissue and can see the antigens of the cancer. So antigens meaning what kind of recognition patterns the cancer cells have to be recognized by the immune system. And then we can really give the information to the immune system. Wow, that's a cancer cell. This is how a cancer cell looks like, and you have to be active against these cancer cells. And if the cancer cells, and that makes cancer very, very difficult, it mutates always. So if cancer is mutating, giving new information to the immune system, you can just adapt the vaccine. Say, well, cancer is mutating, it's hiding. You have to change your weapons like this. So. And you can use also other RNA technologies like CRISPR, for example. CRISPR RNA can cut off gene expression. That's also something very interesting. Or CRISPR RNA can have an enzymatic function of RNA. siRNA can cut gene expression. MicroRNA gives a lot of signals. And you can use the messenger RNA, as I told you. So RNA can do a lot. So, and that's the vision we have, as I told you. So cancer might be a kind of disease you're not anymore dying of, because you can always engage the immune system. That the cancer cells, they always have to hide themselves or to do something against the immune response. So cancer is growing because cancer has nothing to do, it can just grow. But if you can entertain the cancer cells, that they can't grow anymore, that they have to defend themselves all the time, therefore you can live with cancers, and cancer is not really infecting or implementing the organs which are like lung, like pancreas, organs which are necessary for your life. It's kind of entertainment. You have to entertain cancer, then it can't grow anymore. The other thing is Alzheimer. We don't know exactly what happens in the brain, but the older we get, the more issues we have in our brain. And the thing is also, well, maybe you can also interact with that, with the brain cells, to stay healthy. If you see really what makes Alzheimer happening in the brain, you can really defend it also using RNA molecules. And the nice thing in the end, and that's a really utopic thing, maybe also aging, because something happens to our body, we age. Maybe you can also implement like this, that the aging is somehow slowing down a bit more. Or wrinkles or things like that, you know, that's cosmetics, but I don't know. But maybe something, as it's a kind of information to the body, and the body needs information, how to prevent aging, for example, what you can do against it. That can also something very interesting happening. Maybe it's, I wrote it 10 years back, I wrote 2030, maybe it's 2040, but I think this is a really high chance that it will be like that. You can also implement the aging. Of course, we all die. It's clear you can't prevent death, but again, maybe you can die in a kind of healthy condition. Yeah, I think that's really. So I'm really optimistic that we all see this coming up for ourselves, because many companies are now involving China and all the other countries, and a lot of new technologies will coming up like that, and we have a lot of more knowledge about receptors, about functions in the body, and it's the easiest way to target those factors is the RNA technology which is available. So therefore, very bright futures. Thank you very much.
Thank you. I have one question, or maybe two, very quickly, because I remember, I have to ask this because that's, I think, I believe, very interesting. You showed us the picture with Elon Musk. And that's a situation when you have to convince someone who is already convinced because they know something's going on, that there is a business, there is an opportunity. But in the start, I remember you told the story that you presented in front of the renowned scientists, and they told you what?
It's nonsense. Yeah, when I was just a young boy coming from the university, sitting here in a panel talking about the RNA thing, people really don't believe me. And there was a Nobel Prize winner sitting in the first row, and he was really standing up in front of me and saying, it's bullshit what you tell here. It's completely bullshit. What you say against this, you know, it's getting red in my face. I say, well, it's not, I see it because I saw it in mice. I injected mice. I saw it in a living animal that it works. And this guy says, indeed, no, it's bullshit. It's unstable. It can't, because everybody thought RNA is really a molecule which is not usable for any treatment thing. And the thing was really because of the instability they have seen and they have observed, but it really didn't really dig deep enough to see that RNA is the first biomolecule and it's the most stable biomolecule what we have.
That was very interesting, yeah. And this is a message to young scientists to be kind of stubborn, yes, in pursuing your goals. And second example, I remember, this could be the message for startuppers again, because I remember you told later on you've been invited by DARPA in USA to present something about these new discoveries, and then happened something interesting. Yeah. You remember?
Yes, of course. Of course I remember. Well, DARPA is the military agency of the US. So I really didn't know what it was, then I went to Washington, to the defense ministry, really to the defense ministry. You have to go to a lot of security controls. It's really amazing. And I was thinking, wow, what's going on here? And then I saw the DARPA officer with our project. I had a phone call with him, but I saw him now in person, and he had an uniform. So he had really a uniform. He was a military guy sitting there, and I was sitting here on Putin's desk. You know, he was sitting a long way apart from me, and I was telling, you know, how good we are, because I knew how I have to pitch on a positive way, how good we are, what went well, and there's a, you know, as I'm decorating, you know, we can revolutionize and things like that. And he was really looking very, very angry to me and then he was really putting his hand on the table, throwing the hands on it, saying, stop the bullshitting. Stop the bullshitting. I really want to know what went well, because we at DARPA, we fund only high-risk science. We want to really see the risks, and then we can speak about the chances. But I want to see the risk, what went well. I want to really tell me the truth here, because also for me, for my knowledge, I want to invest money that you solve the problems. It's a solving problem things here. And I was somehow flipping and say, okay, yeah, we have some production issues, impurities, for example. It's not easy to control. We have to do more controls like that. Anyhow, we just have animal models. We have to use clinical trials. We have to get the right models in clinical trials. Cancer is a very tough target, but vaccines we can't do because vaccines are all available. So it's a really issue. And say, well, let's talk about it. How we can fix this. And this was something, it changed my life somehow, really to talk also about the negative things and really see how you can solve the negative things, especially to an agency like DARPA. I've never seen this before because the German and the EU funding is always, you know, well, yeah, milestone, 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 everything went well, everything is good. And that's a different the flip side thinking. And this I like very much, that you really do the high risk things, talk openly to the officer, and if things are not going well, then I have trust and he will support me, because this is exactly what he wanted. They want to see really the edges of the technology. They want to see how far technology can go, because it's very important for the military to know the impact of it. And you can only know the impact if you see failures. So that was very, very interesting. A good teaching to all of you. It's important to cope with failures and to learn from failures and to see the edges, how far you can go with the technology.
Okay. Thank you very much, Holger. You will have again chance to talk or listen to Holger afternoon in the session of final session. Thank you very much. And we are now starting those separate sessions. That was the message actually for startuppers. Never trust the guys who don't ask for problems. Okay, if they ask only for money, that's something wrong with this. Okay, so now we split into two rooms. The startuppers to Enterprise B here. We continue with Ahmed. Hello, Ahmed.
We're going to give a couple of seconds for people to just disperse and go to their own sessions before we start. Is there a clicker? Why it's not working? How do you reverse it? Perfect. Okay, let's get started. Good morning, everyone. I hope you've been inspired by the wonderful speaker who was here, giving some really provocative thoughts and some great ideas. I personally agree with some of them. I disagree with some of them, and I will tell you why. Vaccines are not always the solution. And before we get to the vaccines, let me introduce the talk today, which is about the challenge and the promise for modern drug discovery. And why we're talking about modern drug discovery, what has happened that is launching this modern drug discovery. Before I tell you that, I would like to tell you that I'm the clinical data science team leader at Sano, which is a wonderful organization here in Kraków. And my team is also here today to present as well and tell you they're part of the deal from drug discovery point of view. What happened, the event that changed our world in the last few years is COVID-19. If you take a look at this picture, you will see clearly COVID-19 is the biggest workload. You see global pandemic, you see people looking at COVID test, correct? You see people doing contact tracing down there. You also see drug discovery. You also see drug repurposing and drug repositioning showing up to the surface. Okay, so what is this happening? Well, how is this happening? Well, clearly, going back to the point about the vaccines take years to develop. So COVID could not wait. I don't know anyone in this room or anyone in this building who has not been impacted by COVID, whether they contracted COVID, whether they had a friend who contracted COVID, whether had a friend, lost a friend or a family who been impacted by COVID. So to wait for vaccines to develop over years is not a luxury we can afford. So now we have to think of ideas outside the box. Outside the box is drug repurposing. That's what I'm here to tell you today. That's the modern drug discovery from my humble point of view. And I would like to highlight this. So let me give you an interesting definition of drug discovery. And I would like to give credit to Dr. Putzalik here, who allowed me to utilize her slide from a couple of days ago. If you think about drug design in one hand and what it entails to do the synthesis and to discover new molecules, to identify the specificity of these molecules, and to see which molecule would target which type of gene, which type of drug target, and how these targets can be bound with the drugs and the molecules, and it takes years for clinical trials. And then after that, before you even get to the clinical trials, we have to think about what it means to test in silico, what it means to test, to do the molecular docking and all these things, and also testing on cells and animals. Those tiny little poor bastards, we love them, but we don't want to put them through the test of of drugs that we don't trust or we're still working with. And that process takes 8 to 12 years. 8 to 12 years. We don't have 8 to 12 months to save our population from COVID-19. So let alone 8 to 12 years. On the other hand, drug repurposing with AI. Well, clearly, drug repurposing saved the day. And there are stories here that I'm going to show you from the real world of big pharma that drug repurposing has stood up. And it could have not been done without the use of technology and the use of AI. Well, clearly, we have generic drugs that came out of the IP and people can use. These drugs have been aspirin, for instance. If you think about you want to ask me what kind of drug that's been repurposed, aspirin has been notoriously famous for all kinds of repurposing. And then there are added values. And then you basically hear the next step, instead of testing on these animals, you design this 3D model of organoids, which is Dr. Putsalik is an expert in, and all this process takes between 3 to 5 years versus 8 to 12 years, okay? Now I'm gonna tell you, how is this related to AI, okay? AI now getting this really terrible buzz about AI taking on the world. AI is taking away jobs. AI is going to make people cognitively impaired, and all these issues, which I absolutely agree with you guys for the most part. So there are issues also about specific AI issues related to you generate content, you don't know the authenticity of this content, but it looks so impressive. You cannot imagine when you look at something, you cannot imagine that it could be false because it sounds so authentic. And also the facts inside whatever has been generated is horrible. The impact of these facts on our science and our discoveries and our drugs and all these things that people go look into literature and trying to identify the next molecule, the next target, and then imagine this science could be done by ChatGPT kind of engine, and then people publish fake results, and you see these results being propagated, and people use these results and try to spend money on clinical trials, and then they end up cannot even reproduce the results that have been done because they're basically fake. And then ethical standards. When you do research, you have to comply to the ethics. Not everything that you can do, you should be doing. So ethical standards are very, very important subject that must be observed when you launch a study. Especially from an AI point of view, you have to comply to some sort of an IRB board or some sort of a committee in your organization. What are the research agendas to address these issues? We have to think about basics of producing science. We have algorithms today that have been around for 50 years, classification algorithms, clustering algorithms, association rule mining algorithms, network algorithms. We also been designing ground truth we have not been paying attention to, which is stored in databases and ontologies and so on. So we have to really make sure that we have ground truth to counter the fake facts. If we have these ground truth, we can be able to, without anything to compare against, we have nothing to hold on to. So anything can fly if you don't have anything to keep you on check. So here, ground truth is also very important. And then basically have to address the ethical issues from committees that enforces the production of science. I said this example a couple of days ago. I was reviewing a paper for one of those specialized journals in biomedical informatics. And the authors were designing an algorithm using graph neural network, which is basically producing drug combinations that cannot be explained. So I sent my review to the authors and they said, can you explain? They said, we can't. It's a black box. So you're giving me drug combinations that I cannot see how they are being produced, and you want me to go spend money in the lab and waste public money and resources and years in clinical trials and come back and tell me, oh, I'm sorry, I could not, there was just the technology having some sort of hallucination. Unacceptable. So clearly we have to remedy that. So the policies to be able to remedy this, we have to design algorithms to detect fake science. We have to do fact checking. We cannot do it manually. we have to design tools that can actually detect the facts using this ground truth. When you lean on the ground truth, you can actually identify a fact from something that could be a question or a hypothesis or a fake idea. And also designing algorithms that can actually take a look at the black box technology. One of my really hope to do in the next few years is to actually embed these large language models with an explainable module that can actually tell you which articles have been used for what purpose, how these articles have produced features, how these features came from what publication, from what source, embed the metadata. So you can actually know what part of the experiment came from what source, and then you can trace it. But that cannot be done by a human. Now going back to the drug repurposing, I want to remind you that drug repurposing is not a new technology. It's not something that has been used today. COVID-19 highlighted it, hence the word modern drug discovery. But before COVID, Keytruda, for instance, Merck designed this drug earlier in 2014 for advanced melanoma. And now this drug has been the money-making drug for Merck in the last decade or so, and now they're basically trying to recycle it for or repurpose it for all kinds of cancer-related disease. I have personally analyzed some of the experiments. I was working at Merck earlier, and I analyzed the experiments, the repurposability of these plates, and I was ranking them to see which ones that should be moved forward with different configuration parameters, which ones should be trashed. So that's one example. Second example, Pfizer discovered Paxlovid. Also, this is for COVID-19. This is a drug that was been around before the pandemic, but now it's been repurposed and being sold under Paxlovid. Gilead, also remdesivir. Remdesivir was also a drug that was available before the pandemic, and it actually excited the world because it was giving us hope during the time when vaccines were unavailable. People basically used remdesivir for some sort of releasing or treating some of the symptoms of the drug. Now it's been used for COVID-19 and SARS-CoV-2. So these are basically examples of drug repurposing. How do we do drug repurposing computationally? Now I'm going to give you some basic algorithms. There is something called network medicine. Network medicine is the study of the connectedness of disease and the symptoms of the disease, the treatment of the disease, the genetics of the disease, the biology of the disease, the drugs that treat certain disease, the comorbidity of the disease, and the targets of all of that together combined. This is what's called network medicine. Network medicine, when you take a look at this big fuzz of all these entities, you have to analyze it from community's point of view, you have to analyze it from pathways point of view, you have to analyze it from patterns point of view, and this is how you can actually discover some sort of a way through a very large network medicine. I've been lucky to leave my mark on COVID-19 drug repurposing algorithm. Been followed by a wonderful scientist who was actually my former mentor from the Network Science Institute in Boston, Massachusetts, Dr. Barbasi, who also published a wonderful paper in 2021. And then we continue to publish, we continue to advance our drug repurposing algorithms, my team and I. And then we're actually now pitching this idea from COVID-19 into a large-scale workflow that can look at any kind of drug, any kind of disease and address it from a biological point of view. Now we've been very lucky to have Dr. Putsalik with us. She is an expert in breast cancer, and she's also designing organoids. So we shifted this workflow into breast cancer drug repurposing to make it affordable, to make it personalized. So we can take away the cost from Keytruda, which is the cost of Keytruda, or the price for Keytruda is really hefty for average people like myself cannot afford, God forbid. So these are the algorithms, and I want to mention that we are able to advance these algorithms to solve problems that have not been solved today, like breast cancer. How are we using large language models? Okay. We actually, in the previous, before large language models, we never used large language models to design these network algorithms. What was missing really was to identify relationships between a drug and another drug. If two drugs are interacting negatively with each other, they cannot be combined. If two proteins are interacting positively, that could actually be something really interesting that kind of inspire new science or new drug. So what we're looking at right now, we're looking at using the engine of ChatGPT or large language models to ingest them with content that we approve. We design our own attention model. We don't use the pre-trained model. We use the content that we believe that will be sufficient with the help of domain experts. And we can basically then, at that moment, identify relationships among these entities that we're interested in, a disease and maybe a comorbidity of that disease, or a drug and maybe another use of this drug if it points to another drug target, and so on and so forth. But clearly, the domain expertise here are essential. We cannot do this without the domain expertise. I am talking here from a place of being very humble, but also I feel very lucky. I have a team that can actually do the computational work. I have a team that can do the in silico work. I have a team that can do the lab work, the drug testing and the 3D modeling organoids and all that. So I feel like we are very in a very lucky spot that we can offer our insights to anyone who wants to repeat this experiment for another disease. We're happy to collaborate, but we're also looking into the next steps for commercialization. So now let me stop here, and I would like to ask if you have any questions. I know I went so fast. Yes. Generic drugs are essentially out of the patent. Yeah. Because we're actually targeting publicly available data from public databases. So we don't want to get into, again, the idea is to find affordable drugs that can be personalized. So if we're looking at drugs that are, yeah. Okay, if we're looking for drugs that are already under patents, the cost is not going to be affordable. So the idea of looking at generic drugs that are publicly available that people can use and then test in the lab with the domain experts, that takes care of the affordability part. Natalia?
Yeah, I have one question. Because of course, generic drugs, they cover many different therapeutic areas. Mm-hmm. So are you maybe interested in particular areas of therapies, like oncology or
at this moment we're
system, etc., etc.?
At this moment, we're actually investigating drugs, generic drugs for breast cancer, especially for breast cancer. We're in touch with the National Institute of Health in the United States to see how we can get their support. But right now, we're essentially looking at the breast cancer as one significant health problem related to women, and the entire society being impacted by that. We want to make it affordable. We want to make it personalized. We want to be able to manage this disease not only from the disease point of view, but also from the comorbidities, because the comorbidities could be diabetes, could be cardiovascular, could be depression, and so on and so forth.
I understand. Thank you. And are commercial aspects also interesting for you, or you are looking only from the scientific point of view?
Clearly, the next phase for us right now. We've been contacted by some investors, but we wanna be careful. We don't wanna make a rash move. But we're definitely open to serious talks with serious investors. I have my business partner sitting in front of me here. I would be very happy to take this outside the talk.
Okay, thank you.
Any other questions? If nothing else, I would like to present the next speaker. If I can do that. Come on. Okay. Okay. So who is the next speaker? Yeah. Hello, Dominik. It's a pleasure for me to introduce you here, please.
Hello, everyone. My name is Dominik Lipka. Today we have a possibility to hear amazing speak of Ingmar about the RNA revolution. And to be honest, I cannot imagine better introduction to the SyVento than his speak, because SyVento want to be a part of RNA revolution. Basically, SyVento is a CDMO company that want to provide very holistic approach for the development of RNA-based therapy through the development of delivery system. Basically, SyVento is how we do this. We are working in 4 platforms in which we provide specific services to deliver the mRNA-based therapy to the clinic. The first platform is genetic platform, in which we try to synthesize the RNA based on the construct that was developed by our partners, by our customers. Of course, we can support our partners here, for example, with the choosing the best capping system or to provide the best quality raw materials. But basically, in this platform, SyVento is working more as a CMO than CDMO. Second platform is related with delivery platform. And this is where our expertise is the biggest, because we work in this field for several years. We work in this platform from very early stages when we support our partners, our customer in the development of formulation for mRNA, but not only for mRNA. We can help them with the scale-up process. We can help them with production. We can help them choose correct formulation based on their application. We can help them with choosing the best ionizable lipids, because we also work on the very broad library of ionizable lipid. We even try to now implement the AI for this rational design of ionizable lipid. And why this is so important? Ingmar told you that mRNA is highly unstable. And if you want to put it into the human organism, it needs to have a package. And the best package that we have now is lipids nanocarriers. And SyVento tried to develop the best lipid nanocarriers as it is possible for specific therapies, for specific drugs. Third platform is related with manufacturing, because we don't only want to help our partners in the discovery process, in the development process of their drugs, but we also want to help our partners with the production. And this is very important, because we can produce both drug substance and drug products, and at the end do the fill and finish and deliver to the clinic vials with your products. And the last platform is related with the drug development expertise. And in this platform, our people in our labs, in our offices, help our partners with the development of analytical methods, validation of those methods, but also we can support our partners in the preparation of the EMPD documentation. Basically, we want to deliver products of our partner to the clinic. So we try to provide end-to-end services, and this is especially very important for small biotech company who very often cannot afford to have their production, to have the production of drug substance, then production of drug products, or they don't have such a sophisticated analytical equipment. We try to help them to have to realize their dream about the drug. And of course, as we mentioned, our biggest expertise is related with the delivery system. Of course, we work with mRNA, but not only. We work with siRNA, with antisense oligonucleotides, but also with small molecules. By using that, what also you mentioned, for example, we can develop formulation with small molecules, with the drugs that are already known, but with different capabilities. So we can, for example, encapsulate in one liposomes 2 types of drugs and have some synergistic effect that could be completely new compared to the single drug. What is also very important, and I mentioned it at the beginning, we work in different phases. We can start working with our partners from the very early process development, when we support you in every stage, when we try to support you to choosing correct formulation, to choose correct assay, but we can also support you in the development process, scale up. If you have already formulation, then come to us, and we try to implement the same process for example, in lab that are very similar. Basically, they are the same for the process that are in the GMP production, which is very important. What is also very important, because we are located not so far from here. We are located in Skawina, and probably most of you is not aware that in Skawina, like 4 kilometers from here, we are now finishing building, building construction is already finished, but we are now working with validation. There will be located a GMP facility that allow us to produce mRNA as a drug substance in GMP standard, and also drug products, LNP liposomes, as a drug products. And we can also do fill and finish. And what is also very important, in the emergency situation that we have in the COVID pandemic, we can switch to because we are, our production line are designed for the production for the clinical trials. But if there will be need, we can produce up to 35 million doses of the mRNA-based vaccines. As we mentioned, we support our partners in process scaling. We try to develop process in the laboratory that are very easily scalable for the production. And of course, all that things cannot happen if we don't have a nice analytical lab. We have really nice analytical lab that develop analytical method for characterization both drug substance and drug products. In our labs, we've got possibility to characterize drug substance, mRNA, according to the latest draft of United States Pharmacopoeia. And I believe we are the only one in Poland who can do this. And the same for the drug products. Beside that, we've got biological laboratory, and we can perform some basic biological assay like MTT test for measurement of cytotoxicity, but also protein translation. And we've got partners, and we are still looking for a new partners because we have a lot of projects related with in vivo testing of products that we are developing together with our partners. Thank you very much. I hope that I fit in the time.
Thank you so much. Please don't go away. Do we have any questions? Okay, Adam has a question, and I will get back with you. So 2 questions. Yes, please.
Yeah, thanks for talk. First question, more related with commercialization process. What do you think, what is easier to commercialize, RNA sequence or nanoformulation, or in both, because RNA is not stable, so in terms of commercialization, it's terms of commercialization RNA sequence with formulation.
Yeah, it's a very tough question because there is, I believe, not easy answer. But if you want to have RNA, mRNA based product, you need to have both. Nice developed sequence of mRNA and also formulation. So it's both very, very important.
Are you using some sophisticated targeting on these nanocarriers, like some ligands or peptides, which...
So simple answer, yes, but we do targeting in 2 ways. One, passive targeting, I will say, which is based on the composition of the lipids in the LNP, because they can also help to target a specific organ. But we also try to work with active targeting using, for example, small molecules, peptides, parts of antibody. But to be honest, for CMC documentation, it is also a nightmare. So basically, we prefer passive targeting.
Thank you very much. Very interesting talk, of course. Just a question for the big animal in the market. It's Acuitas. Everybody uses their technology, so it's a kind of gold standard. My question is, do you have any data in comparison with Acuitas technology in animal models? The other question is, Acuitas has a very strong IP position. What is about your IP position, and are there infringements with Acuitas patents?
So basically, for now, what is also very important, we work as a CDMO company. Of course, we've got some lipids, I say, on board. And if you compare to the Acuitas or to Moderna formulation, in animal models, they work better. For example, if you choose, I don't know, luciferase as a reporter gene on EPO, but at the end, we are not the final user of the product. But we believe that the chemical structure of the lipids are completely different to the Acuitas or to the Moderna lipids, so there shouldn't be any problems with the patent.
Okay, thank you.
Do you have like any experience, and what is?
No,
like...
we are not focusing on the vaccines. To be honest, most of our project is not related with the vaccines, but for example, with the rare disease, for protein replacement therapy, personalized therapy also, not only vaccines.
Okay.
Thank you so much. Let's give our speaker a Wonderful. Now, I would like to introduce Dr. Adam Sułek. He is going to tell us how he is predicting the properties of drug targets using AI technologies.
Thank you for introduction of myself, and actually I wouldn't imagine better introduction regarding the talks which we already hear, because from both sides. Because if we are looking on proteins and mechanism in organism, DNA stores some information, then RNA translated to the protein. So a step before engineering the proteins, like in my case, we should use RNA. And also formulation is very interesting subject, because we could also consider proteins as nanocarriers in drug development. I'm Adam Sułek, and I'm working in Sano. So my perspective is mostly scientific development. But today I want to focus on some business aspect of my work. Have you ever wondered about the journey, how introduce new drug to the market? I had this opportunity to take part in every step of clinical trials. First, we need some proposition of new drug. So we are using some established method like molecular docking, QSAR methods, but still in silico methods are only theoretical, so without real evidence. So we need to test this in the lab. However, testing in the lab gives you some insight, but without applicability. So then we need to test it on living organism. But yeah, it's still hypothetical, even though today's talk we all heard that it's very insightful, and after positive animal test we could believe that this drug should work. Still we need some test on other models like 3D organoids. And we could consider zero clinical phase as something before clinics. This experience, because in my previous work we get some drug into clinical second phase, teach me that there is the gap between preclinical and clinical trials, and this gap consists of cost and time, because there was two numbers, 5 to 8 and 8 to 12 years. And it depends how we consider, because if we have good proposition of in silico, then probably it could take only 5 to 8 years. But if we are struggling few times in this first step, give bad propositions, so it's iterative process instead of one-shot prediction. And I like this quote that AI, because now it's very popular, but it's not a standalone tool that we are just using AI. It requires human expertise and collaboration. And here lies the pivotal question. What evidence do you need to confidently approach your manager, boss, or investors to give you money to your model? Because of course, everyone could say that my model gives me 80%, 90, 95% of accuracy. But, first and foremost is that we are testing on data which was never seen before. But the great idea would be, first tell me what this experiment will produce without knowing the result of experiment, and then we will check if it's really works. So it would be the best proof of power of your model. But you also consider whether your prediction is useful on the market demand, because many times scientists are predicting some value, but it's not with market demand. In my case, I'm trying to predict melting temperature for proteins. To build you the draw what is melting temperature, during the Comp Biomed conference, we were into brewery, and there was very hot space where this beer was produced. It requires high temperature, and this process requires enzymes, which are proteins. So engineering proteins which can stand higher temperature, it's effectively better for production in this case. And also the experiment, if you plan in your project, sometimes are costly. So you should consider whether it's in your scope and budget. In case of melting temperature of proteins, is not very cost experiment. And the introduction of new drug to the market, the time are doubled every 9 years. So if this phenomenon is called Ehrum's law, but nowadays we can, and as you can see, each year there is less and less drug introduced to the market per $1 billion. And recently we observed this trend has been reversing, probably because better information for decision. And I believe that better access to data, better access to model like I'm using in my work, large language model for proteins are key assets nowadays. So, and also you should remember that this market is quite different than, for example, medical application or other product which can be rolled out every year. Here you have to be very patient to have initial product. And this leads me directly to the next point. Groundbreaking solution, very famous in scientific world, it's AlphaFold. And the reason why it's so popular, because they winning the CASP competition. And this is exactly how I described before, how good is your model. Because first, there was experimental 3D protein structure, and then the Google DeepMind, it was according their prediction. The value is invaluable on the market, but nowadays we can use software as a service from NVIDIA using this model, large language model for proteins for your purpose. It could be prediction of 3D structure, But in our case, we can predict the temperature of the proteins, melting point of proteins. So your model is only as good as your data. So in our expertise, we filter data from the same type of experiment regarding temperature, and we obtain a promising result with mean error of 4 degrees of Celsius. Why this is better? Because most scientific approach are trying to predict whether it's thermostable or not. So predicting in this range, you can see that the prediction could be very wide up and above this line. So it's very not accurate. In our case, we obtain, it depends on the protein, but better result we obtain for this not thermostable. It's large language model for proteins are more accurate in this range. So we need to improve our model with active learning. So it means that we require new experiment in both, in validation of our model, as I mentioned. First, your model should predict something, then you have to prove that in the lab. But also having more data means that it's better to train our model. And the commercialization is actually during this talk was said that one thing could be commercialization of turbo-stable sequences, but also building this model could be part of some licensing product. And also here I mention different use case of thermostable proteins. It could be considered as drug vehicle. Small peptides are used as antimicrobial drugs. So there is still many use cases how we can use thermostable enzyme with our model. Thank you for your attention.
Questions for Dr. Adam Sułek? Okay, we're running a little bit late, so if we don't have any questions now, we can take them outside the room. Thank you so much again. Now I would like to introduce my next customer, Piotr, correct? And he's going to tell us about an interesting type of radiation. It's called synchrotron, if I can pronounce it correctly, on how it can be used for drug discovery. The floor is yours.
Okay, thank you. My name is Piotr Ciochoń. I'm the Industry Liaison Officer at Solaris Synchrotron. And today I will tell you a little bit about the methods that can help you in the drug discovery and development processes. So first things first, Solaris is the interdisciplinary research center located even closer to here than SyVento, because we are only 700 meters away, which operates the first and so far the only synchrotron light source in Central and Eastern Europe. I will get into the details of what the synchrotron is and what methods do we have available, but let me just stress that we are part of quite unique group of institutions. There are only 14 such facilities in Europe and only around 40 worldwide. Okay, so what is this synchrotron? The synchrotron is a particle accelerator. It's the large device which forms an electron beam, then accelerates it to nearly the speed of light and keeps it enclosed in a circular orbit in the so-called storage ring seen here from above. Unlike CERN, because we are a bit like CERN, but not really, we do not smash this beam into anything. So we do not smash it into another beam, into any targets. Instead, we are focused on keeping the beam alive for as long as possible, circulating in the storage ring, because every time the direction of motion of the beam is changed by the bending magnets, they emit radiation, so-called synchrotron radiation, with some unique properties. This radiation is then extracted out of the storage ring and directed to the beamlines shown here by the arrows, and to the end stations where our scientists perform experiments. This is a large device. It has the circumference of 100 meters, and we have quite a few of beamlines. But why do we need such a large and expensive device? Well, it comes down to two things. First of all, synchrotron radiation covers a very wide range of frequencies of energies, so from infrared to visible light to soft and hard X-rays. And second of all, it's very intense or very bright, as we say in physics. There's some difference, but I will not get into this. Anyway, this effective intensity is orders of magnitude higher than the standard laboratory sources, which is especially visible in the X-ray regime. For our users, this translates into the possibility of performing rapid, precise, high-resolution measurements with very low detection limits and high quality of experimental data. And while those two things are true for all the techniques that we have at our disposal, we also offer access to several methods which require synchrotron to work. This is the plan of our facility. In the heart of it is the storage ring, as you can see, and we have quite a few beamlines available. In fact, there are 7 beamlines which are available right now, with many more in different phases of project, of development or construction. And each of these beamlines uses only a small part of this wide electromagnetic spectrum that I showed you before. That's why each of the beamlines can be treated as sort of semi-independent laboratory. This means that we have access to a wide variety of different techniques, from spectroscopy techniques, which allow us to study the composition of material, the chemical environment of elements and their oxidation state, through microscopic methods, which allow us to image the matter on different scales, very often with chemical sensitivity and the ability to probe interactions. And finally, diffraction or scattering methods, which allow us to study atomic structure of matter, order, disorder, phase transformations. I will not get into details here about all these methods because we don't have enough time. Let me just show you a few examples of how they can be used, especially in the life science sector. The first example is use of high-quality FTIR data to perform automated tissue classification in biopsy samples. This has been actually done by the team of our Siri beamline, and they were able to develop a machine learning model with quite nice accuracy and all the other important parameters based on this high-quality experimental data. The second example concerns X-ray multimodal imaging, which allows us to study the chemical composition and the distribution of different elements and their chemical environments, for example, inside living cells. Here you can see the example of distribution of potassium and arsenic inside a cell, and we can even say that the arsenic absorbed by cell has a third degree oxidation state free. So these are two examples, but there is more, because in addition to the synchrotron itself, we also operate a cryo-EM center. We offer access to 2 state-of-the-art cryo-EM microscopes, Glacios and Cryos. Both are equipped with bioquantum filters, direct electron detectors, and more importantly, we have a team, an experienced team, which can provide access to all the fundamental techniques associated with cryo-EM. So from electron tomography to single particle analysis to microcrystal diffraction. And here you can see an example of application of cryo-EM from this year, where our team or users at our facility were able to solve the structure of TRPM8 channel with atomic resolution. And I should have actually mentioned before that solving these structures doesn't require crystallization of proteins. So this is one of the big advantages of cryo-EM. Okay, so you know a bit about what we do, what methods do we have, and how can you access our services. There are 2 main routes. If you are an academic user, the access is based on the open calls for proposals. Usually we have 2 calls per year in March and in October. And the proposals are evaluated by independent international committee, which can grant the access to the chosen beamline in the time frame of 6 to 12 months. We do have some rapid or test access options, which allow you to access the beamline faster, but we do require publication of the obtained results. If this is something that's interesting for you, all you have to do is go to sun.synchrotron.pl and apply for the chosen beamline. However, if you are interested in different types of access, if you're especially from industry, we do have a dedicated structure at Solaris, Industry Liaison Office, which I'm the head of, which serves as a single contact point between industrial partners, researchers, administration, and legal teams, and everyone else at the facility. So our services are fast, so we can typically provide access within 1 month of the initial inquiry. Confidential, we offer full confidentiality of the results. We are ISO certified when it comes to data management, data security. And our services are also comprehensive because we can complement our techniques with the methods available at our partner facilities. If this is something that's interesting for you, all you have to do is write an email to industry.solaris@uj.edu.pl, and we will take care of the rest. And specifically for cryo-EM, we have 2 predefined set of service packages. The first one is sample characterization, which is a screening plus package, which allows you to assess the quality of the sample, to preliminarily characterize the proteins that are in your sample. And based on these results, we can recommend a go or no-go decision to the next phase, which concerns full structure reconstruction. In the first sample characterization package, we keep the data and we keep the grids, the frozen grids at our facility. So you can use the pre-characterized grids for the full structure reconstruction in the second service package. We are also very well connected in the European innovation landscape. So we are members of LIPS, which is the League of Accelerator-Based Photon Sources, which groups all the synchrotrons and free electron lasers in Europe. We are a member of CERIC-ERIC, which is a distributed research infrastructure of Central European facilities. And we are also coordinating and participating in quite a few Horizon Europe, Horizon 2020 projects. Here I mentioned the project Cylinda, which aims at increasing the industrial use of synchrotron radiation. And with this, I would like to finish. Thank you for your attention. If you have any idea of collaboration, please send an email or add me on LinkedIn. Thank you.
Thank you again. And do we have 1 minute for questions? Any questions for Piotr? I have one. Affordability. How is your technology affordable so you can do drug discovery that for people here can afford?
That's an interesting question. So, like, first and foremost, our goal is to advance science. So we are not profit organization. So we are a research organization. So this is our first goal. So any scientist can come to us and propose a project, and if this project is good, if it's evaluated positively, the access is free of charge for the scientist. So it is affordable. It is affordable. Thank you. If you want to use it for proprietary research, then that's a different story.
Fantastic. Thank you so much again. Our next speaker, Justyna Popiul from Jagiellonian University. And she's going to tell us about her discovery of these novel inhibitors to design cosmetics so everybody can be beautiful. So hold on and listen tightly.
Okay, thank you. Hello, everybody. My name is Justyna Popiul. I would like to present you results of our studies on novel melanogenesis inhibitors for the usage in cosmetic products. The research was financed by National Center for Research and Development within LEADER 11 program. Hyperpigmentation disorders, recognized as dark spots, patches, or changes on the skin, result from an abnormal amount and or inadequate deposition of a pigment melanin in the skin. They include, among others, freckles, melasma, age spots, and inflammatory and hormonal pigmentation disorders. They are significant aesthetic and medical problem and also negatively affect mental health. Currently used therapeutic methods of hyperpigmentation disorders are insufficient. In addition, substances used in the therapy are characterized by side effects such as irritation or allergic reactions, as well as limitations in preparing formulations. Sorry. In light of these facts, there are significant premises for searching for new melanogenesis inhibitors. Thus, the aim of the project was the identification of a novel cosmetic ingredient for the enhancement of the treatment of hyperpigmentation disorders by inhibiting their formation. We focused on a selection of innovative active substance with confirmed efficacy, optimal physicochemical properties, and a favorable safety profile. In our studies, we used reference in vitro models and modern research equipment. Intellectual property protection was also a priority. We designed, synthesized, and tested several dozen of cinnamic acid derivatives. Compound A111 proved to be the most promising. Due to limited time, I would like to present results obtained for this compound. The inhibitory melanogenesis activity was evaluated using 3 in vitro models: mushroom tyrosinase, mouse melanoma cells, and pigmented reconstructed human epidermis. Studies performed with mushroom tyrosinase showed that compound A111 shows strong inhibitory activity. The obtained IC50 values were comparable to kojic acid used as a reference compound. Kinetic studies showed that test compound acts as a mixed type inhibitor. Studies using B16F10 mouse melanoma cell line allow us to demonstrate that compound A111 inhibit melanogenesis process stimulated by alpha melanocyte stimulating hormone. Before starting experiment, we conducted studies which aimed to exclude the cytotoxicity of compound against used cells. Before beginning the activity studies using pigmented human reconstructed epidermis, the conducting of safety assessment was also necessary. The aim of the study was to demonstrate that any potential decrease in melanin content does not result in tissue damage. Compound A111 was found to be safe at all tested concentrations. In studies conducted using a pigmented reconstructed human epidermis, we demonstrated that a 14-day treatment with compound A111 resulted in significant decrease in melanin content with no impact on tissue viability. We also conducted safety evaluation of obtained compound. Using cell lines of human origin, we excluded cytotoxicity of compound against skin cells such as keratinocytes, fibroblasts, and primary melanocytes. Compound A111 showed to be also deprived of hepatocytotoxicity and neurocytotoxicity. In vitro safety studies using FRITI-free NRU phototoxicity test and micronucleus test allowed us to exclude phototoxicity and genotoxicity of compound A111. In microplate format Ames assay, the mutagenicity of compound A111 with and without metabolic activation was also excluded. In the next step, with the aim of performing studies in future application conditions, we incorporated compound A111 into oil in water emulsion. Then we tested the safety and activity of formulation. The obtained results were very interesting. Using pigmented reconstructed human epidermis, we showed that formulation inhibit melanin formation in epiderm. After 14-day treatment, the melanin content in epidermis was 62% of control. In addition, the formulation was also deprived of phototoxicity. It was showed using epiderm model. Additionally, using EpiSkin model, we proved that compound A111 incorporated into cosmetic formulation permeates through EpiSkin. Thus, it is possible that upon skin application, it has the potential to penetrate deeper layers of the epidermis and inhibit the melanogenesis process. Our invention is a subject of Polish patent application and European patent application. Currently, we are looking for a business partner for future development and possible commercialization of our invention. The next step of development of our project is conducting in vivo studies which confirm the efficacy of a compound. The novel dynamic acid derivative with anti-melanogenic properties could become a novel ingredient of depigmenting or bleaching cosmetic products. Thank you for your attention. I'm going to answer each and every question in a coffee break. Thank you.
Thank you so much. I'll take this one. Are they made for men too, or just for women? No worries. No, I was just making a joke. I would like to introduce the next speaker, Joanna Kajaminska, if I can pronounce the name correctly, from Rolniczy University. And she's going to tell us about the secret of superfood, is known as kale, and how this can actually prevent chronic disease, non-communicable chronic disease. The floor is yours.
Thank you so much. My name is Joanna Krzemińska. I am PhD student at Department of Human Nutrition and Dietetics, Faculty of Food Technology at Agriculture University in Kraków. I want to presentation you entitled how kale as superfoods and with high potential of health prevention against the development of chronic non-communicable disease. And chronic non-communicable disease such as diabetes type 2, cardiovascular disease, osteoporosis, thyroid disease, gastroenteric disease. This is very important for human health because according to the World Health Organization, are responsible for then more 63% deaths and worldwide abilities. The cause of chronic non-communicable disease to include consume of high processed foods, excess red meat and in the diet, unhygienic lifestyle, lack of physical activity, too little intake of fresh vegetables and fruits, increasing environmental pollution, and reaching for stimulants such as alcohol and cigarettes. And why kale? Kale is a superfood because inside have a lot ingredients of high bioactive effects of our body. Brazilian vegetables, including kale, have a lot of amount of bioactive compounds, so highly recommended for consumers to improve health in chronic non-communicable diseases. Inside our kale, we find phenolic content, carotenoids, glucosinolates, fiber and protein, and we find a lot of vitamins such as E, A, B1, B2, ascorbic acid from C, vitamin C, and K. What's more, inside kale we find a lot of minerals such as irons, calcium, magnesium, potassium, and manganium. What's more, very important questions is presence sulforaphane inside these plants, because this chemical structure cause anti-cancer properties, which has been proved studies research. And biofortification with iodine in the form of iodohinolines future improve the nutritional value of kale better. Our research testing the influence of effect biofortification with different type of iodines in plants, such as kale, red and green form. And our results, the present study clearly shows the biofortification with iodohinolines can increase the content of total iodine inside these plants, total polyphenol contents, antioxidant activity, and carotenoids, sometimes macro and micronutrients. These slides we can see type of form iodine, in organic form potassium iodate, and organic form 8-hydroxy-7-iodo-5-quinolinosulfonic acid, and the second form 5-chloro-7-iodo-hinolinol. We using foliar testing in the cultivar red boar and olden boar. Red boar, this is the red kale, and older boar, this is the green kale. And we observe significant improve iodine, polyphenols, carotenoids, and antioxidant activity inside these plants. The next steps our research was analyze the influence of thermal treatment on the properties of kale biofortified with iodine. Sometimes different type of foods is not possible eating in raw forms, so we must do using heat treatment. And the level of bioactive compounds depends on the way it is cooked and how it is processed. So the most common technique used by consumer is the heat treatment of food products, including kale, so which inducts several biological, chemical, and physical changes inside these plants, such as kale. So this study analyzed the biofortification effects and it changes in the total iodine, total carotenoids, total polyphenols contents, antioxidant activity, chemical compositions such as fiber, protein, ash, and other parameters. In kale, two cultivars, older boar and red boar. It was no biofortified with iodine. It was the control, and biofortified three forms: potassium iodate, positive control for us, iodohinolines two forms, and we know that, however, we found a significant decrease in the content of total iodine, polyphenols, antioxidant activity, and ascorbic acid under steaming, blanching, and boiling process. So, kale is very, very healthy food for us, and we have a good influence our body because decrease no communicable disease, and we recommended consuming no Bio45 and Bio45 kale in raw forms for salads and lunch dishes. As kale best retains its nutritional value, including iodine, under these conditions. And while consumption of kale, due to this wide range of health promoting properties, and we can reduce the incidence of chronic non-communicable disease, given the most optimal cooking techniques. And suggested dishes we produce, we made our kitchen. This is the soaps, so creams, salads, crepes, smoothies, cake, ice creams, dessert, puddings, pancakes, pesto, and other tarts and other dishes. And I invite you to together work. Our Department of Human Nutrition and Dietetics, it conducts scientific activities and on the assessment of the diet and nutritional status of different population groups, and impact of a diet and dietary components on health, in vitro research on cell lines, in vivo research animals using mouse and rats, and population studies. So we encourage collaboration and joint research project, research and development, educational activities, and popularization of science in the field of nutrition and dietetics. So the offer is aimed at dietetic companies, clinical nutritionists, other medical institutions, students wishing specializing in human nutrition and dietetics, and produce of foods or dietary supplements. I can tell us that we produce actually dietary supplement using Bio45 with iodine vegetables such as lettuce, potato, and kale, two cultivars, and we produce other ingredients of diet where we can edit these Bio45 vegetables. And thank you so much. Sorry, author funding and bibliography. These results presented above presentation were developed by a lot of people. Our department and this research was financed by National Science Research, and it's all. Thank you so much for your attention. Any questions?
Thank you so much for such a healthy talk. Any questions? Yes, sir.
Thank you. So my point is, how different is this vegetable from the others? So have you validated and compared to the other vegetables to claim that this one is particularly different from the others? Because they are similar, so the family of these vegetables have the same properties, so and you are really focusing on a single one.
Thank you. Very interesting question.
I'm not sure I understand everything.
Basically saying why kale?
Why kale? Because we had to level these ingredients inside: protein, fiber, macro and micronutrients, and polyphenol content, carotenoid as ascorbic acids. Then, for example, which analyzed earlier, spinach, lettuce, and other vegetables. So I know the kale is not popular for human consuming and health, but this is very, very interesting vegetables with this advantage.
I guess you guys have a date to talk about.
Oh, thank you. Thank you so much.
Absolutely. What is the word? What's the Polish word for kale? Jarmuż. So if you're not Polish, say after me. Jarmuż. Yarmush. Yarmush. Okay, now eat your kale. Alrighty, thank you so much. Thank you. Okay, we have the last speaker, but not the least, Andrei Shaviets from Cancelleria DBS, and he's going to tell us how we can get early access to new treatment. Thank you so much.
Good morning. My name is Andrzej Siwiec, and I'm attorney at law and partner at DBS law firm from Kraków, a boutique law firm specialized in legal services for life sciences sector. My presentation will be a bit different than the previous presentations, as I'm not even able to talk about scientific details, but I would like to present some legal framework that allows us to provide medicinal products to patients as soon as possible. So the topic... That's important too. Yeah, I think we also have to focus on not only on the scientific part of the product, but also, how to provide product to the patient. And so the topic of my presentation is early access programs, how to accelerate patient access to new treatments. I present a bit information about medicinal product life cycle. Also, I'll focus on a specific period, which I call a gap, and then I present you two possible ways to provide medicinal product to patients during this period between the end of a clinical trial and but before the marketing authorization is granted. From clinical development to the market. Now on the screen you can see more or less the medicinal product life cycle, and at first we have this first phase of clinical and first non-clinical and then clinical trials. Then we have a second phase, which is analysis of the pivotal clinical trial results and preparing the registration dossier, marketing authorization application, and then whole marketing authorization registration process. Once we have marketing authorization granted, we can launch the product in the standard way and supply product to the patients in the standard market way. And then we have, of course, work on pricing and reimbursement and other aspects of commercialization. And I would like to focus on this second stage, which is launch preparation. And during this period, at this stage, we have kind of a gap, because once the clinical trial is completed, we can no longer provide patients that were enrolled in the clinical trial with a medicine. And because we have not yet the marketing authorization granted, the standard supply is not possible. This period, it takes 2 to 3 years if everything works smoothly. So it is not a short period, and we have positive results of the pivotal clinical trial, and we cannot supply the product to the patients, and it is especially harmful in the case there is no other approved treatment for the patient. So it would be good that the patients can benefit from this treatment if they have no other therapeutic option. And for that purpose, there is a kind of a legal institution of so-called early access programs, and we can use them to accelerate patient access to the investigational medicinal product after the clinical trial is completed. We have 2 types of these programs. One is compassionate use, and second option is patient basis access. They are already implemented they are provided by the EU law, but the details then have to be implemented by national regulations. The idea of compassionate use program is that it is a cohort program for a group of patients. It is usually pre-approved by authorities, and we cannot use it for to deliver the product to a specific patient, but we have to establish a specific protocol and enroll patients according to this protocol. And second option is a named patient basis access, and we can use this option to supply product which is unauthorized to individual patients upon request made by a physician and on its responsibility. It may be a better way, for example, for rare diseases where there is only 1 or 2 patients in a country and there is no possibility and no sense to set up a cohort compassionate use program. And to give you just some country examples and present that it is a bit different in each EU country. In Poland, we do not have option to set up a compassionate use program, but we can supply medicine according to the named patient basis access upon a request of a physician, and only in case if there is, of course, no other treatment option on the market, and the product has to be delivered in order to save life or health of a patient. In Belgium, the rule is a compassionate use program for a group of patients, but in specific urgent or life-threatening situations, there is an option to use named patient basis access. In Sweden, we have both options. For example, there's an option for pre-approved compassionate use program for a group of patients, but also you can obtain special license granted by authorities to a specific patient or to a specific hospital, so all patients in a hospital. And at DBS, we have particular experience in supporting our clients in navigating through the different legal landscapes in EU in establishing early access programs, but also in all launch preparations for medicinal products. But we also advise our clients on other aspects of pharmaceutical market and other life sciences projects. So I thank you for your attention. If you have any questions, I strongly encourage you to contact me. Thank you.
Thank you so much. Very useful information. And any questions? Yes.
I have a question with regard to the scope of presentation which we have already, that many of you says that personal medicine is real and is our future. So how do you feel, what will be the following the law changes with regard to personal medicine? Because we can consider this that is a kind of a individual treatment, and with the regard of the allowance for using such products. How do you think that will the law rules will adopt to the real future of biotech?
Yeah, I think the key is the key are pricing and reimbursement regulations, because personalized medicine is not cheap. So payers have to accept the high price of the treatment, and the fact that there is no general reimbursement decision for a specific drug, but each time they have to accept the insurance, the coverage for the specific patient. So I think if the governmental authorities, insurers, other payers, accepts the high amount of money to pay, the other regulations will follow.
I have one short question. So you mentioned a compassionate use, yeah, and it's applicable, for example, for rare diseases, orphan diseases. Yeah. And do you have maybe any examples of compassionate use compounds which were accepted by the regulatory? And could you elaborate a little bit more about the whole procedure? And are there differences between member states in terms of compassionate use? Thank you.
Yeah, in fact, I'm currently working on a approval, distribution, and early access program for orphan drug. However, compassionate use programs are usually not the most suitable way for them because you have only 2, 3, 10 patients in a country, so it's it's difficult to set up the whole compassionate use program with protocol, with involvement of authorities for a small number of patients if it is not paid. So yeah, it's difficult. The compassionate use programs are more frequently used in France and Belgium, as far as I know, because I'm Polish lawyer, but I work also with colleagues from these countries. And for other countries, from my experience, the preferred way is patient-basis access, which is different in each country, but it allows, in fact, to supply product in most of EU countries.
Okay, thank you.
Thank you.
Do we have any questions? Any other questions for? Well, let's give our speaker a hand. Okay, the closing remarks. I must say, I'm absolutely delighted to see the diversity in the speech, the speeches that we had, the diversity in the science that's been done, the diversity in the type of treatment that has been offered, also the diversity in the science related to the drugs and the use of technology of the drugs. To give you example, use of technology, we actually seen AI methods. We used computational algorithms. We saw network medicine. We saw in silico work. We saw 3D organoids type of work. We also saw interesting technologies using the accelerator. This is also something that was truly interesting to me because when I think accelerators, I think about these dark matters going and you have to look for them in tunnels and dark places. I did not imagine that there will be a drug discovery type of technology coming from this place, which is really, truly inspiring. And we also saw different methods on how to address the treatment. Some of these methods were related to drug repurposing, as I presented earlier. Some of these methods are also by our honorable keynote speaker with the vaccine development. Some of the other technologies are also looked at the novel discoveries with the traditional methods in labs. So this is also a very rich type of discovery. And let's not forget that we also had three different gifts, in my opinion, to this very interesting session here. One of them is the significance of eating healthy, how eating healthy can actually help with the chronic disease. Thank you so much to Joanna for the introduction of kale. And also we saw, what's that? Yarmush, yes, I will not forget that one, Yarmush. And the other one was also very interesting. I would like to talk about myself. I always feel ugly. I feel like I wish somebody designs these cosmetics for me so I can look beautiful. So well, where's our speaker who is designing these cosmetics? So I would like to thank her personally for that. And then we also had the regulatory part and the policy part and logistics of how to make the medicine accessible. We concluded by the Andre's talk, and I feel like this is really a very comprehensive meal, very heavy. You can really truly enjoy. But I would like to mention something to conclude this talk with. One of the things that I wanted to talk about, but I knew I was running out of time, so I did not say anything. The significance of drug repurposing today is essential when it actually comes to disease that we don't know anything about. For instance, Alzheimer's has been mentioned today. During my time, my tenure at Merck shut down some clinical trials related to Alzheimer's. As a matter of fact, if I remember, after that, their program was being disbanded entirely from the company because they could not identify one single drug that would actually treat Alzheimer's. And then recently, in the last year or so, the NIH, National Institute of Health in the United States, were actually calling researchers from all over the world, including people from Europe. As a matter of fact, Sano was one of those people who wanted to participate in this call for proposals about investigations of drugs that actually failed in clinical trials. This is how significant drug repurposing is, to be able to take a look at drugs that have failed, and then how you can boost these drugs. You can maybe, if they failed for efficacy, you can combine them with another drug that would actually make them more potent. And if these drugs are poisonous or whatever, toxic, you can also apply these other computational medicine tools to do the QSAR that Dr. Sułek was talking about here, on how you can remove the toxicity and maybe design a new drug. Also, the significance of identifying drug alternatives, to identify the drug alternatives from a medicinal chemistry point of view. You can take a look at drugs and look at the similarities of these drugs and how we can analyze, how we can predict the behavior from the structure. So I would like to leave us here with this talk, that the significance of drug repurposing today, especially for disease that we don't have anything to defend against, like Alzheimer's, for instance. We're all getting older. Is getting, and I heard the question today of how we can stop us from aging. We just can't. We just need better drugs, and we need better health, better cosmetics, so we can always look beautiful and look young. Thank you so much for being here today. It's an honor to present present the speakers today, and it's an honor to have all of you as my audience. And enjoy your break. Thank you so much. Yes.
Bravo to our host. Thank you.